So I welcome you all in today's class. It will be on gene cloning. So you can have an organism cloning also, like the dolly, that is this first sheep that was cloned, and then SKUAST also cloned one Noori by Dr. Riaz Shah, and that was a hit. Um, the animal cloning is different from gene cloning. Okay, there what we do, we take a cell, a somatic cell. Okay, yeah, you can say an ovary cell, enucleate it, and then put a somatic cell inside the nucleus of somatic cell, so that the organ that grows is the actual clone. So we're not dealing with that, we're dealing with gene cloning. So there are various aspects of gene cloning and there are various prerequisites of gene cloning. So what you require for gene cloning is, number one is your vector. Okay. Number two is your insert or gene that you want to clone. Okay. Three, you require a host. Okay. So 4, you require restriction enzymes. 5, you will require ligase. This is also an enzyme. So these are the prerequisites of cloning. So vector is a part of DNA that we use to transfer our gene of interest. Okay. In entomology, we call vector. A vector is kind of an insect or someone who transmits the disease, the virus, the bacteria from one plant to another. But in this case, the vector is a DNA. In this case, a plasmid DNA that will help in transferring our gene of interest. Okay, into, or what we say, how to clone it. Okay, so vector is nothing but a circular DNA, in this case a plasmid DNA. Okay, any circular DNA cannot act as a vector because there are some attributes of a vector that and the prerequisites of a vector. If these prerequisites are not met by the DNA sequence, by the circular plasmid DNA sequence, we do not call it a vector. Okay, so the first and foremost property of a vector, okay, is that it should have an origin of replication. In this case, REC. Origin of replication is important entity of a vector. If a vector does not have this origin of replication, it will not be dispensable. What I mean by dispensability is that, for example, if you have a cell, bacterial cell, okay, and you have this vector inside, okay, if this vector does not have an origin of replication, in next mode of reproduction, say binary fission or whatever, 2 cells will be formed. Okay, if this does not have an origin of replication, this will not replicate, so one copy will stay as one copy. So this one copy will either go into this or this. Say for example it goes into this. So after one cycle, you have 2 cells and one cell has your vector. In the next cycle, this will divide again into 2 and this will again divide into 2. So you have now 4 cells, 4 daughter cells, and again, since this is not replicating, it does not have an origin of replication, it will only be here in one cell and one will be devoid of this vector. So after millions of, or say n cycles, you have millions of bacteria, but only one will have this plasmid, okay, or this vector. So we do not want that. We want that our vector should be dispensable, that because when you're cloning a gene, you need a lot of copies of the gene for future experimentation. You cannot have only one copy. So what we do, we put an origin of replication here so that this vector divides into two, okay, replicates, what we call as DNA replication, and one will go in one and another one will go in another one. So here now this will also have, and here you will have. So after n number of cycles, if you have millions of bacteria produced from one bacterial cell, each and every cell will have a plasmid because of this origin of replication. So origin of replication is an important entity of a plasmid. It is an important attribute of a plasmid. If a plasmid does not have this origin of replication, this plasmid is not dispensable, so that means you'll lose it. Second thing that a vector should have is a marker. Okay, it's not this marker. Okay, it's marker for selection. In this case, an antibiotic resistance gene. For example, if this vector has an ampicillin gene, okay, the product of this gene, what it does, if this plasmid is inside a bacteria, okay, and this has an antibiotic resistance gene, in this case ampicillin. So what it does, this ampicillin gene or ampicillin resistance gene, it produces beta lactamase. This beta lactamase has a property that it breaks the beta lactam ring of ampicillin and renders it inactive. So now if somehow this plasmid goes into a cell, E. coli cell, okay, initially if this E. coli cell does not have this plasmid, if you grow this E. coli cell on a medium containing ampicillin, this cell will not be able to grow, so you will see no colonies because this bacteria is not resistant to ampicillin. However, if now you are taking this plasmid that has an ampicillin resistance gene, you put this in the bacteria, you put this in the bacterial cell or E. coli in this case. In lab we use again the DH5 alpha strain, okay, it's easier to maintain, so it's a modified strain, it's easier to work with. So now you put this vector inside this E. coli. Now you grow it on an ampicillin plate, LB medium that has ampicillin, or medium that has ampicillin antibiotic in it, and you grow this inside. You will be able to grow this because now this bacteria has Irshad, can you put your mic off? Everyone of you, just put your mics off. So, so now if this bacteria, this vector has an antibiotic resistance gene, and this vector is put inside an E. coli or a bacteria. Now we grow this E. coli on a medium that has ampicillin. This bacteria will be able to grow because now it has ampicillin resistance. Okay. So this is how we'll be able to know when we transform our E. coli, when we clone a gene, we transform our vector into the bacterial cells. Which bacteria harbors our plasmid will be known because they'll be able to grow. If the bacteria does not have a plasmid, it will not be able to grow. So this marker for selection is important because once you take your gene, okay, you clone it, you clone it, you put it in okay, you took a gene, you cloned it in the vector. Okay, now you put the vector inside the E. coli cell through a process called as transformation. Okay, transformation is this process through which a bacterial cell can take inside a foreign DNA. Okay, if one bacteria helps in putting a DNA fragment into another bacteria, that through a pilus, it's called as conjugation. Okay, and if a virus does the same, it's called as transduction. Okay, we do not want to go in detail. So simply, if we mix our cells with the DNA in presence of calcium chloride, okay, or DNA gets into the cell. The transformation practical will be doing. So, but you have millions of bacterial cells, you put millions of copies of this plasmid inside these, and there will be a chance that not all these bacterial cells will harbor a plasmid. Some will not accept a plasmid, okay? So if after transformation, you have just these two bacterial cells that have incorporated the plasmid, but these two have not. So when you grow this mixture on a plate, LB plate, and this plate has ampicillin in it, only this and this bacterial cell will be able to make colonies. Why? Because they have this vector, and the vector has ampicillin resistance gene. This and this colony, the bacterial E. coli cell will not be able to grow, they will not be able to make colonies because they do not have resistance against ampicillin. क्योंकि हमने मीडिया में we have put ampicillin antibiotic in the medium as a selection marker. Okay, so second important attribute of a vector is that it should have marker for selection. Third important thing that a vector should have is that it should have an MCS or a PCS. It's called as multiple cloning site or a polycloning site. This is the site that is rich in your restriction recognition sequences. Like if you go to the previous class or the previous YouTube video on restriction enzymes, we know that restriction enzymes are palindromic. We know that they are recognized by, they have a sequence, what we call the restriction site, and these are recognized by a specific restriction enzyme. So we know that multiple cloning sequence has a number of restriction sites. Okay, what we have to do, this restriction site, this multiple cloning site has restriction sites, like it has site for BAM-H1, okay, EcoR1, HIND3, okay, KPN1, and many more. Okay, there are like hundreds of restriction enzymes. So you have these many restriction sites here in the MCS or the polyclonal site. Because now what we have to do, this is our vector. Okay, we have to cut it at the multiple. This is the multiple cloning site here. Okay, we cut it, and this is our gene of interest. Then we ligate this gene of interest inside the vector. That's our goal. And put this vector inside a E. coli cell. Right? This is our goal. This is how we clone. So we'll cut the vector at the multiple cloning site. Okay, and the ends of the sequence of our gene of interest should also have this multiple cloning site. For example, I have made a vector for you. You see this DNA sequence. Okay, this is a DNA sequence. Okay, this is a plasmid DNA. So hope it's visible. This is a plasmid DNA. Okay, this is an open circular plasmid DNA. Okay, and if you see, I have kept some registration sites here. Okay, this is the multiple cloning site. Okay, this multiple cloning sites have 10, 15 recognition sequence of registration enzyme, different recognition enzymes. They are overlapping sequences, so that in a smaller portion you have a lot of you have many registration sites. Okay, so for example, this site is GAATTC. GAATTC is the recognition sequence of anyone? Anyone? GAATTC is the recognition sequence of? Can you be a little bit louder? Can you be louder? Yes. Who is it? Oh, good. So it's the recognition sequence of EcoR1. So what we have to do, this multiple cloning site ensures that हमारा जो vector है, हमारा जो gene of interest है, that goes inside the vector. Okay, it forms a plasmid. So what we do, so this is your restriction enzyme. Okay, these are called as molecular scissors. So, okay, are you able to see? Okay, so this will cut here, GAATTC, it will cut here, right? Then another cut on the same sequence, because they are palindromic, will be made here. Okay, so these sites have broken two phosphodiester bonds. Now you have these hydrogen bonds in between that is keeping them together. So what we do, we'll cut these like okay. So this vector, circle vector, we have cut at a multiple cloning site. Okay, I'll keep it aside for a while. Now this is your gene of interest. This gene of interest we want to clone inside this vector. So the prerequisite is that you will use a restriction enzyme. You have 15-20 restriction enzymes. So how will you select the restriction enzyme? So the restriction sequence that you will select should not be anywhere in this sequence. If your restriction site is between this sequence, because what you have to do when you design primers to amplify your gene, you put the same restriction site at the end of your primers, so that when it amplifies in a PCR, you have a gene of interest flanking with your restriction site. For example, here you see I have kept GGA, okay, GAATTC again, okay, there also it was GAATTC. So you know that I have this gene of interest, okay, I want to clone in the vector, okay, so I'll use the multiple cloning site to open up the vector. I will cut it with the restriction enzyme. Here EcoR1, I use EcoR1. I have to ensure that EcoR1 site is not anywhere between this gene, because what we have to do, we have to also restrict this fragment with EcoR1. Why? Because I have kept EcoR1 at the flanking ends of this gene, right? So you have a vector, okay, that I have cut with EcoR1. Okay, sorry, a vector that I have cut, and the insert that I have also cut with EcoR1. Now the ends are compatible. If you cut one EcoR1 DNA here and another EcoR1 DNA, you cut another DNA in say United States with EcoR1, they'll have again the compatible ends because they recognize the same sequence. In this case, if this is your insert, okay, now you have on one end you have again GAATTC. Can you see? Again you have GAATTC right here. Again, okay, because we are digesting the insert also with the restriction enzyme that are used to digest the vector, so that same restriction sites are at the end of both because they are compatible. So we'll cut it like this. Okay, so this is the end you got. These are called as compatible ends, and the ends they have produced are sticky ends. So GAATTC again. Here. Is it visible? Okay. Now you see you have an insert that has compatible ends with your vector. How? Now this is your insert. I'll keep it like this. Okay. This is your vector, right? When you mix the two and add ligase enzyme, what will happen? This will form A will pair with A will pair with A, T will pair with A, T will pair with A. Sorry, A will pair with T and T will pair with A. So they'll form these hydrogen bonds, and then with the ligase will connect this the phosphodiester bond and this phosphodiester bond. So this is your one end. This is your another end. Okay, so you see here you again have right. So in this way, now you have a vector that has your insert. And how did we do that? We restricted it with a restriction enzyme that was in the multiple coding site. So multiple coding site is a site that is rich in your restriction recognition sequences, and it is a site where we put our gene of interest. Fourth property is that your vector should be small in size. Okay, otherwise it will be just trying like to get an elephant inside the house. Okay, so that's why we use the quality of a good vector is that it should have small size. It's one of the attributes. So that's why the multiple cloning site here is overlapping. That means in a smaller region you have more number of multiple clones. If you Google multiple cloning sites of a vector, you will see that if this is the recognition sequence of equar1, recognition sequence of say BAMH will be like this. So they'll have an overlapping region, so that you can make the vector size smaller. So smaller vector, better to control and better to clone things in it. So another thing is that the vector should be small in size. Then you have another property of a vector is that it should have a marker for recombination. This marker for recombination is here. This marker for recombination lies. Okay, I can say multiple cloning site lies in the marker for recombination. Why do we require marker for recombination is that For example, you took a mixture of plasmids, okay? So this is a mixture of plasmids, okay? Right? And some have this insert. You took a, okay, see what happens. You took a plasmid, okay? You restriction digest it, okay? You mix it with an insert that has the same restriction sequences at the end. You ligated it to, and now you have a sequence like this, right? But the thing is that if you see a vector, okay, this is your insert. Some of the insert does not get incorporated, okay, does not get cloned. So, but when you have restricted a vector with a restriction enzyme, you see the sequences that they produce, okay, they are compatible ends. They can again religate or re-anneal, right? So chances are that when you put them in a solution of ligase, okay, ligase will bind them together back, right? There's a chance, okay, because the ends are compatible. It's a vector, okay? Or either an insert can go in between. Both things can happen. So that's why we require marker for recombination. Because for example, say these are the vectors, okay, and say in two of the vectors you have your insert or gene of interest, but others religated. They are empty vectors. When you transform these these vectors in the E. coli, okay, and then you plate those E. coli on an LB plate, all of these will be able to grow because you have ampicillin here, right? And all of these vectors have ampicillin. So you have vector, 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 vector. Now you do not know that which of these have your gene of interest, right? Because here you have only two of these vectors have gene of interest, but you do not know because they will also grow, these will also grow. So you are confused. So you have a mixture where you have empty vector and then you have gene of interest. Okay. So to overcome this problem, we use a marker for recombination. So marker for recombination, for example, in this case when we use LAG-Z. LAG-Z is a gene that breaks X-gall. Okay. X-gall is a compound, okay, or a molecule. When X-gall breaks into two molecules, one of them is blue in color. It is a blue color. Other one is colorless, right? So if we see now this situation, you have a multiple cloning site. Let's draw it separately. So you have, this is your multiple cloning site, this is your marker, right? And you have another condition also, you have marker and you have this MCS. This way, this LAG-Z, this is LAG-Z, right? LAG-Z marker. If we have not digested this with restriction enzyme, this LAG-Z is functional. Okay? And if we take a restriction enzyme, we break this LAG-Z because multiple clone site is in between the LAG-Z. But when we ligate it, it again ligates back. That means we are rejoining the gene again back. So that means it will again be functional. So if a vector is there, you restriction digest it because, and then ligate again it will be functional because they are undoing the function. You are ligating it back. So if the vector is empty, no insert has been incorporated between, LAG-Z will be functional. And if an insert comes in between, this is an insert, okay, it gets cloned between your gene. So now your LAG-Z is from here to here एंड फ्रॉम हियर टू हियर, राइट, हमने उसको तोड़ दिया, इसके बीच में हमने जीन ऑफ इंटरेस्ट डाल दिया. सो नाउ we have two scenarios, okay? One is that we restriction digested it, okay, and then it got ligated back. So this will produce functional lag Z. Good. In this case, when we restriction digested it, we cloned our gene of interest between that, right? Because we are adding or insert. These are multiple cloning sites on both sides, and this is your lag Z part 1 and lag Z part 2. This will not be functional because now you have an insert in between. That's a big gene, so it will make this gene dysfunctional. So lag Z is not functional here. Now there are two circumstances. If this goes in the E. coli cell, if this goes in the E. coli cell. So this time what we do, we plate them on ampicillin, right? Because this is a part of a vector that has ampicillin gene here. This is part of a vector that has ampicillin gene here. Okay, so we grow it in ampicillin, both will grow, right? But here what we do, we add X gall, a chromogen like ampicillin in the medium. Now both these cells will grow, right? And they will form a colony because all are resistant to your ampicillin. But now here what will happen, this colony or this vector which does not have an insert, its lag Z is functional. It will produce lag Z, functional lag Z, and that will degrade the X gall in your medium. Once the X gall is degraded, it will give blue color because one part of that what we call the product of X gall, it breaks into two parts, like as I told, okay, the one part gives a blue color. So these colonies will appear blue because of a functional lag Z. But blue colonies are not our colonies of interest because we know that they don't have a gene of interest. If they would have a gene of interest, their lag Z will not be functional. When lag Z is not functional, they will not be able to degrade X gall. When X gall is not degraded, they will give simple off-white colonies. So that's why we require marker for recombination or marker for screening. This marker helps us to differentiate between those vectors or those vectors that are empty, that do not have a gene of interest, we do not require them, and those vectors that have our gene of interest, right? Good. Another important property of a cloning vector is that it should have high copy number. Copy number means, oh boy, Any questions till now? Fossil Fayaz.
Yes, sir.
Any questions?
सर जो ये कलर आता है ब्लू कलर, ये पूरे इसमें आता है मीडियम में कि ये सिर्फ कॉलोनीज में आता है?
सिर्फ कॉलोनीज में आता है. दैट्स वाई. मीडियम में आता है तो फिर कैसे पहचानते हैं? ये सिर्फ कॉलोनी ब्लू हो जाती है.
सर
वही.
ये इनको एक्सट्रीट नहीं करते हैं ये प्रोडक्ट?
नहीं, ये कॉलोनी ना, ये एक्सक्रीट नहीं करते हैं. It gives blue color to the colonies only. एक्चुअली जो मीडिया, जो एक्सगॉल होता है वो आपके तो मीडिया में है ना? तो जहां पर मीडिया के ऊपर होती है वहां पर वो मीडिया में से वो उनको ब्लू कलर में कन्वर्ट कर देता है. एंड इट्स लाइक ब्राइट ना, इंडिगो ब्लू. सिंपल सीधे दिख जाता है. इट्स डे एंड नाइट. यू कैन गूगल. Okay, so this meeting is in continuation of our previous meeting and the previous YouTube video. Okay. So if I share my screen, can you see? Can you see my screen?
Yes, sir.
Can you see these displayed?
Yes, sir.
Okay, this is your blue-white selection. So you see how blue colonies are and how white colonies are. So our interest is in white colonies because these are the colonies that will have our gene of interest. And blue colonies are empty vector, religated, so they will not have our gene of interest. Okay?
Yes, sir.
So, and another property is that it should have high copy number. So this lecture is in continuation of the cloning lecture, and here we are studying the properties of your cloning vector. So here another property that we talk about is high copy number. That means one E. coli cell will harbor a lot of plasmids. That means not one plasmid but many plasmids. So two different types of plasmids cannot stay inside one cell because of the process we call as plasmid incompatibility. Okay, and but similar kind of plasmids can stay in a cell. Okay, and like for example, if we there's a vector called as PUC18. Okay, we call this vector, this is plasmids, this P is small, C is capital, is the University of California developed this plasmid number 18. So it has 500 copies in one cell. Okay, so we prefer that our host in which we clone or a vector should have the property that it should have high copy number, because if you have one copy number, if at the end of a cycle you have say 1 million cells, so you'll have 1 million plasmids. But if you have a copy number of 500, that means each cell will have 500 plasmids. That means 1 million into 500 is around 500 million plasmids at the end of your experiment. So you'll always prefer that. So why this happens is we make this the host, okay, faulty in replication. Matlab when replication occurs, okay, rolling circle replication or whatever, D loop replication. So for example, it's a rolling circle replication. This is a plasmid, okay. There's a nick, okay, and now the condition is like that. This is not in your syllabus, but it's not bad if you do not if you know this. So now replication occurs here, okay, it goes like this, and this strand is peeled off like banana, okay. This again moves, this is again peeled off until and unless you have the hole, and this one recircularizes. Sorry, this one recircularizes and second strand is synthesized. So this one is now two strands. What will happen? This will stop replication here, okay, because the process that there are proteins called as riproms, okay, they bind to this site so that replication does not refire. We call it as refiring of the origin of replication. So if we mutate those riprom proteins, okay, that bind here, it will recognize it. It will not recognize that it has. We have to stop here. So what will happen? It will go again, keep on going. So this will peel off. This will again keep on going. So this will peel off, and at the end again you have two plasmids, and this will peel off and make another plasmid. So by refiring of the origin of replication, you have multiple copies, like mini copies, like 500 copies in your bacterial cell. So these are the properties of some of the properties of your cloning vector. Now next step is your preparation of the insert that we studied. So you have an insert. Either see, if you want to clone genomic DNA, it's simple. If you want to clone a gene, it's a little bit complex if it comes to eukaryotes, because in case of eukaryotes, in case of, say, for example, in case of prokaryotes, you take the whole genomic DNA, okay, you take the vector, digest it with EcoR1, digest this with EcoR1, okay. So you have so many inserts. Insert 1, 2, 3, 4, 5, 6, 7, and many. EcoR1 will digest, okay, 4 to the power 6 times. The probability of digestion of EcoR1 or recognition of EcoR1, okay, if it recognizes here, it will recognize after 4 to the power 6 bases. But we do a partial digestion, like for a smaller time, or we use less enzyme, so that we get larger fragments of the genomic DNA. So what we do, we got now these fragments 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and then you have vector. So since it's a mixture of vector, you have some millions of copies of vector, okay. You again digest this with EcoR1, so you have like this. Okay, now you mix these two, okay, you take this and you take the vector. We take the insert, this is the gene or the DNA we are going to incorporate, we call it an insert. Insert to vector ratio 2:1. Insert we take more because the probability is that the vector will ligate self-anneal. So but if the inserts are many, the probability is that it will religate with the vector. So you digest genomic DNA. So this is how we make a genomic DNA library. It's sort of cloning the whole genomic DNA of a bacterial cell, say for example, or plant cell. Say in case of, we'll first take bacterial cell. So we took the DNA, digested with EcoR1, take the vector, digested with over EcoR1, mix the two, okay, in the ratio 2:1. Then we add ligase. Restriction enzyme does not require ATP, okay, because this breaking of bond, it releases energy, but ligation, we require ATP. So the buffer that comes for ligase consists of ATP, okay. So we mix the vector and the simple mixture, take 2 microliters of insert, 1 microliter of vector, 1 microliter of ligase, and say 2 microliter of buffer, then add water, and also you should keep the volume of the reaction very low, because higher the volume, more the dilution. So vector and insert will be parts away. But if you keep the volume low, say 10 microliter reaction, it will be good. So you have a vector 2 microliter, sorry 1 microliter. You have insert 2 microliters. You have ligase 1 microliter. You have buffer say 1 microliter, and you have water 1, 2, 3, 4, 5, 5 microliters. So total volume is 10 microliter reaction. Then what you do, the ambient activity of a restriction enzyme is 37 degrees centigrade. So when you restriction digest, you put it in a water bath at 37 degrees centigrade. Ambient activity of ligase is also 37 degrees centigrade, but we do not keep it in at 37 because at 37 the kinetic energy of the molecules is high. For example, so that's what we do. When we do this, we keep ligation at 16 degrees centigrade or 8 degrees centigrade. So simply we take the tube, mix everything, and then put it in the fridge in the lower corner. So the refrigerator that comes, the lower shelf where the vegetables, we keep vegetables, it has a 16 degree temperature. You go up, it has 8, then it has 4, okay, and the freezer it has sub-zero. Now, for example, this is your vector end, and this is, and why do we use 16 degrees centigrade? Okay, we do not use 37 degree for ligation. We use lower temperature, 8 to 16 degree temperature. It is not the ambient activity of ligases, but still it will have some activity. Okay. So now this is the insert. Okay, so this is the insert, and this is one end of the vector. Okay, now they have to ligate, for example. Okay. Now we do the ligation at 37 degrees centigrade. At 37 degrees centigrade, the kinetic energy of these molecules is high. Okay, so they're in random motion. So now I want to ligate it. Can I be able to ligate? No. Okay. Now we lower the temperature 16 degrees. So kinetic energy decreases. Okay. Now if I try to ligate them, they'll be able to ligate. Right. So this is one end, and similarly, you have the vector, and then you have the another end. So it will be ligated like this. So that's why we keep the temperature at 8 to 16 degree for ligation. Once the ligation is done, we keep overnight ligation. Next day we come, we take this mixture, we call this ligation mixture. We take this ligation mixture, okay. Now see what will happen to the ligation mixture. Initially what you have added, the insert and the vector. So you will have a position where you will have a vector, okay. Some molecules will have a vector, and we will have a insert. Good, the thing that we want. In some cases, the vectors would have religated, right? And in some cases, you will have where the vectors are religated, your inserts will be freely moving, right? They'll be in the mixture, right? And now what you do, you take E. coli, DH5 alpha strain we usually use in our lab, okay. You have BL21 if you want to express proteins. There are many strains. This E. coli we have to first make competent, okay. We have to induce competence. Competence is that it should be able to take this DNA inside it, okay. If they are not competent, they will not take the DNA inside it. In normal conditions, what happens, they have competence factors. They are proteins associated with the membrane of the E. coli. So this DNA binds with these competence factors, they have propensity towards them, and the competence factors then help the DNA to move inside the cell. This is the normal transformation that occurs in nature. But here we have to force our DNA into the cell by forcing the cells to become competent. How we do that? We take our E. coli cells, okay, we grow them overnight, 16 hours at 37 degrees centigrade at constant shaking, like we did when we did plasmid isolation. But when we were isolating plasmid, at that time that DH5 alpha had a plasmid inside it. This time its nail, it does not have a plasmid inside, it's empty. It only has a genomic DNA. Okay. So what we do, we take E. coli cells, grow them overnight. Okay, next day in the morning we come and we put 1 ml of this E. coli culture into a fresh tube and keep it for 4 hours constant shaking at 37 degrees centigrade. So in that 4 hours, what happens is your exponential phase is achieved. Okay, competent cells are best made when they are made in when the cells are in exponential phase. If you take the overnight culture for making competent cells, the cells will be in the stationary phase and about to die. Okay, they don't have good competence. So what we do, we take overnight culture, put 1 ml in a fresh tube, put it for 4 hours. In 4 hours, hopefully our exponential phase will be achieved, and we'll take these cells and put it in a 2 ml tube and centrifuge it. Okay, you have to handle them delicately. These competent cells are very delicate. So once you pellet them, what you do, you add 0.1 molar calcium chloride. Okay, then you tap it and mix it. So these cells will be now dissolved into calcium chloride. Calcium chloride is the main chemical that helps in making competent cells. How it does, there's not a proof, but there are two hypotheses how calcium chloride increases the competence. So when we mix the cells, okay, and we have to keep this do the whole process under laminar airflow, and this we have to do under sterile conditions, because contamination should not be there. So, and this all thing we have to do in ice. Okay, we take the cells, okay, centrifuge them. We have the pellet, throw the supernatant, that's the used medium. Then the pellet, we add 1 ml of 0.1 molar calcium chloride. Then mix it again, centrifuge it. This is called preliminary washing. Then your pellet will be here. Okay, throw the calcium chloride. Then again add say 500 microliters of calcium chloride, 0.1 molar, the same solution. Okay, and then resuspend the pellet. We take it to, we resuspend it gently. Okay, you don't have to be harsh, you don't vortex it. So you gently resuspend it until you have your cells here. Okay, now you can keep it on ice for 5 minutes. Now you take this ligation mixture and add into your tube. So you take the ligation mixture, you add it into the tube, and then after addition, you keep it on ice for again say 5-10 minutes. Then what you do, some people keep it for 30 minutes. Okay, it depends upon how your lab does it. So once 30 minutes are over, you simply mix it and take it out, and then you put it at 42 degrees centigrade for 90 seconds. This is what we call as heat shock. We took the tube from ice that has our ligation mixture and competent cells, put it in water bath for 90 seconds. It should not exceed 90 seconds. It should not exceed 90 seconds. So 90 seconds you take it out and put back it on ice. Okay. Now you have a mixture. This is called as heat shock. It increases your transformation efficiency. Okay. Now you take this and add LB medium in it, say 500 microliters. Okay, and keep it at 37 for 2 hours. Okay, here what we are trying to give this bacteria a good, nice, cozy medium, so that the heat shock and the cold shock that they had, they'll come out of it. Okay, they don't die, and the these host cells in which they have the plasmid, they'll be able to grow. So for 2 hours we don't give any selection pressure. We simply put them in LB medium or a sock medium we can use. Okay, it's a highly nutritive media. We can use both. Put them here, and for 2 hours we keep it at 37 at constant shaking. After this, say you have added 500 microliters media. Okay, Now you want to spread it on a LB ampicillin X gall plate, right? LB ampicillin X gall plate, you will pour this solution, but you'll not pour 500 microliters. There's a lot of solution to be added on a plate. So we usually use not more than 100 microliters. So what we do, we have to use all. So what we do is simply spin it. Spinning, what will happen? You'll have a pellet here and then you'll have a supernatant. So we'll discard 400 microliters of this supernatant and 100 microliters we'll keep here. And slowly with the help of a pipette, we'll resuspend this pellet here back and take this 100 microliters and spread it on this plate. Okay. So now what will grow on this plate and what will not grow on this plate? So one condition, as I told you, you have a plasmid vector that will have an insert, okay, that was transformed into an E. coli cell. Another case was your realigned, or what you call religated plasmid, okay, it went into the, got transformed into the E. coli cell. Another case was your simply insert was there, okay, it got transformed into the E. coli cell. Okay. Another case might occur where you have one vector, you have another vector. So you digested this vector with EcoR1, right? You digested this vector with EcoR1, right? Now either an insert will go inside this or they will religate. But it can also happen that this is complementary to this. So they will be able to, because it has the same receptor site. So they will religate and they will religate, right? This will give us false positive results. Because in this case, your gene of interest will be not there, but the plasmid is there. You have a vector, you have a plasmid, again another plasmid here, and it has changed the configuration of lag Z. Now lag Z is not functional, but there you will be getting a white colony. Then you are confused what will happen, right? So these are false positives that you have to take care of. So this will be able to grow on this E. coli, on this plate, and it will have a white colored colony, right? These will grow, but they will have a blue colored colony because of your marker for screening or recombination. This will not be able to grow because it does not have an antibiotic resistance, plus it does not have an original replication, it is not dispensable, so it will die. So these cells will not be grown. Now you have another cells in which you have a plasmid vector ligated to another plasmid vector, and now they are inside a cell and they will give you a white colony, right? So once this is done, next day you spread it on the plate, next day you come, keep this plate at 37 degrees centigrade, okay, and then you have these colonies. You now pick up these colonies, your white colonies, and you again grow them in your 5 ml medium. You take tubes, okay, say LB tubes, okay, with kanamycin, sorry, with ampicillin, all will have antibiotics. Take this colony, the white colony, colony number 1, colony number 2, colony number 3, colony number 4, okay, then grow them overnight. Next day you come and you isolate plasmid in isolation by alkaline lysis that we did the other day, right? And then you check for your inserts. How we do that? When we run these plasmids, how many bands we'll get?
3.
Multiple
3.
bands. Okay, some plasmids give 2 bands, some give 4, some give 5. Your PEGFE that we used the other day gave 3. Okay, so we say always it gives multiple bands. This is the right answer. Okay, so you will have bands here. Okay, that means your transformation is successful. Now to check your cloning, what we do, for example, this is your vector and this is your insert. Okay, this is your vector. Okay, this is one side of vector and this is your insert. Now initially we digested this with EcoR1. Now my question to you is, can we be able to digest it again with EcoR1? Can we be able to again digest it with EcoR1?
Yes, sir.
Yes, we'll be able to digest it with EcoR1. So what we do, we take our plasmid, we digest it with EcoR1 again. Okay. Now how many sites of EcoR1 are there? How many sites of recognition sequence of EcoR1 are there? One is this side, on one side of the insert, and another one is on this side, other side of the insert. So you have two recognition sequences for your EcoR1, right? Because we have ligated, the EcoR1 sites have reconstructed again, because insert की भी sequence वही थी और vector की भी sequence वही थी. So when we digest it, EcoR1 will digest here like this. So, and when we run the gel again, you will have how many bands? How many bands? 2 bands, because this has cut here. This will give one band that will be like this, the vector band, and this will give the band that will be the insert band. You will get 2 bands if your cloning is correct and if your insert is correct, and by this, and the molar ratio of the both will be same, that these both the bands should be of the same intensity. Okay, so that means you have cloned this insert in this vector. Now for example, your vector was, you got a vector colony where the vector was religated. When your restriction digests it, you will only get one band that will be of the vector but no insert. Right? Now you have a condition I just told you, you have 2 vectors, right? They have 2 restriction sites. This will give, this is a vector, this is a vector. So you have now 2 vectors. They have the same size because they are the same vector, because we started with the same vector. So you'll get one band here, and that will tell us this was a false positive. So cloning is confirmed when you restriction digest it, and you will see that you have 2 bands. There is the same molar ratio. One band corresponds to the size of the vector. If you have a control vector, if you restriction digest it, you will have a band here that will tell you that your vector band is correct. It should be at the same molecular weight position. And then the anti-vector will not have an insert, but the recombinant vector will have an insert that will tell that your plasmid has been, your gene of interest has been cloned. Okay. Now next question arises, how we do cloning of a gene. Okay, this was in case of eukaryotes. Sorry, in prokaryotes. Prokaryotes cloning is easy because they do not have introns. However, in eukaryotes you have introns. So that means the gene is larger than it has to be, because there are intervening sequences in between the gene. So what we do here, we do not, in case of eukaryotes, we do not clone the DNA, but we clone the mRNA, because your gene is this, it has introns, it gets converted into hnRNA, it has again introns, then these introns get spliced out, and then you have your messenger RNA that is smaller. So we clone this messenger RNA or the functional gene in case of eukaryotes, right? Or we can use genome DNA for cloning of genes that do not have introns. So that should be all about today's lecture about cloning.